I've been looking forward to this conversation all day. It hasn't been a long day yet, though. Today we're going to be talking about how to regulate the products of cellular agriculture, especially with the focus on fermentation products like milk and egg proteins, but also on cultured meat. Now, Vince, you're an expert on this topic because we already have a lot of fermentation products out there in the market. Can you tell me a little bit about your expertise?
Sure, thank you, Isha. Actually, it's interesting you posed the question in this way, how to regulate cellular agriculture products. It might be a bit counterintuitive to assume that we can influence this, because in the end the regulators will define how to regulate these products. However, we have a say in this. We have a say in this as cellular agriculture innovators, as food scientists, as well as safety experts. And hopefully that will become clear in the course of the conversation. Yeah, I've been involved in a regulatory field on the food side for about 15 years, last 10 years in industrial biotechnology. So industrial biotechnology really is the area that leverages fermentation-based technology as well as recombinant DNA technology to produce enzymes and other small molecules for use in food, for use in animal feed as well, and a number of other applications. And so this is an area that has evolved over time. We've established a safety evaluation methodology for enzymes. I've been involved in that effort as part of the Enzyme Technical Association. And so this methodology has achieved acceptance by US FDA and other agencies as well. So we'll talk a little bit more about that.
Yes. Can you tell us what the main kind of regulation we're working with when we talk about fermentation products?
Yeah, these enzymes and other fermentation products, they've been around for quite a while. We're talking about age-old fermentation products like yogurt, like cheese, like bread. Believe it or not, bread is actually a fermentation product. Think about it. It involves yeast, right, before you stick it in the oven. More recently, enzymes have been involved, have been used in similar processes as well. And these enzymes have been used for quite a long time, well before 1958. And then more recently, of course, biotech-based enzymes. And all of these have been eligible for relatively fast-track approvals by FDA. And FDA is the main, the Food and Drug Administration is the main agency regulating food additives as well as GRAS components.
So you mentioned 1958, so we're going to put up a slide of what happened in 1958. I understand many food ingredients are regulated under the Food Additives Amendment of 1958. Correct,
yeah, this amendment, and we won't bother you too much with details, but this was an amendment put in place in 1958 to modify or amend the Food, Drug and Cosmetics Act of 1938. Back in 1958, concerns were raised regarding the use of chemical additives to food, largely chemical preservatives added to food. So this amendment then defined that any ingredient to be used in food is considered to be a food additive, which is subject to formal approval by US FDA through a so-called food additive petition process, unless that ingredient or that substance is generally recognized as safe or GRAS.
So there's only one path or the other?
Yes, the two are generally mutually exclusive. You choose either or. So we can talk about what differentiates the two. Basically, GRAS is an exemption to formal approval. The developer or the company makes, with help from experts, makes its own determination of the safety of a substance. whether that's a fermentation product or any other ingredient.
So the
Yep.
the company is responsible for reporting the regulation data?
Absolutely, yep. Now, the amount of data that is needed to support the safety of a GRAS substance is exactly the same as the amount of data for a food additive petition. There's minor differences. Food additive petition covers environmental aspects a bit more in detail. But the big difference is that a GRAS determination is the company's responsibility, and in case of a formal food additive approval, it's FDA's responsibility to make that determination. Big difference there.
Okay.
Yep.
So this pathway is for all food products, which are considered food additives. How would fermentation products in particular follow this pathway?
Yeah, I'd say most fermentation products will qualify if substantiated appropriately with the necessary data for GRAS status. So currently, for example, enzymes used by fermentation by microbes generally follow the GRAS determination route. And then companies have the option to file a GRAS notice with US FDA. To date, about 700 GRAS notices have been filed since that program, the GRAS notification program, started in 1997. Of those 700 or so GRAS notices, about 70 or so have been on microbial enzymes.
Oh,
An
interesting.
additional 20 or 30 on other fermentation products as well. But microbial enzymes have made up a significant portion.
Okay, interesting. So it seems to me, because GRAS is the exemption, that it's probably the preferred route, especially because the companies are responsible for collecting the data, and it may be simpler, faster than the food additive petition?
It is indeed, the process definitely is simpler. Like I mentioned, the data burden of establishing GRAS status through scientific procedures, which is what we do today, is just the same as for a food additive petition. However, there's a couple of differences in that the pivotal safety data has to be in the public domain.
Okay.
This can be done through publication in scientific journals. It can be also done by using an expert panel to review your GRAS determination and write a consensus opinion. So that's one big difference. The other difference is that when you file a GRAS notice, you end up filing a summary of the safety determination, of course in sufficient detail for FDA to make sense of it. But it is quite a bit shorter than a food additive petition, which indeed has to contain all of the full study reports, toxicological studies, and so on.
Sorry,
And
which one requires the data to be published?
GRAS.
GRAS?
Yep.
Okay.
But the filing, when you make a voluntary GRAS notice filing, is a much smaller dossier compared to a food additive petition.
Okay.
The review time of a GRAS notice by FDA, which again is voluntary, is only 6 to 9 months, sometimes
Okay.
a bit longer, but can be as short as 6 months. Whereas the review time of a food additive petition usually is multiple years.
Wow.
It has been as long as 5, 6, or even 7 years in some cases. So definitely, if you can qualify the substance that you're making as GRAS, that is highly preferred in order to get to market quick.
And do you think that a lot of these cellular agriculture products, like milk proteins made in culture, egg proteins made in culture, would be GRAS because there are milk proteins in nature, egg proteins in nature?
I would say so, yes. Actually, if we can
Shall
go
we
to the next slide.
walk through the GRAS process?
Yeah, that's probably a good idea. So this here is a set of puzzle pieces that addresses each of the essential components of a safety evaluation. This was developed for microbial enzymes. And as you can see behind me, there's 5 pieces to the puzzle. There's the so-called substance, or in the case of enzymes, it would be the enzyme protein. There is the strain, microbial strain, or cell line, right, in the case of cultured meat. Of course, the manufacture process with all of its inputs as well. And then the underlying safety data, this can be in the form of toxicological studies, which can include 90-day rodent study, for example, as well as genotoxicity studies. And then finally, the exposure. So the exposure really is the worst case scenario consumption, maximum consumption that you can conceivably figure out of consumers consuming food categories that might include or might have been processed with the substance in question. So this was developed for enzymes. It works great. There are some more detailed decision trees actually that we use to support each of these puzzle pieces. So going to fermentation products, cellular egg fermentation products like egg albumin or milk proteins, they essentially are manufactured in very much the same way as enzyme proteins. Mm-hmm. And so I would think that they can follow the same route, the GRAS approval route, as well as follow a very similar safety evaluation methodology. The advantage of milk and egg proteins, of course, is that these are substances that are already in food. They've been in food, for ages, centuries. So from a substance familiarity perspective, FDA will have no issues with that. Of course, all the other pieces of the puzzle, the safety puzzle, will still need to be addressed.
Well, we often talk about one of the benefits of cellular agriculture being the opportunity to modify things or change things a little bit, or for instance, design an egg protein that can whip up more fluffy, for example. How do those little tweaks to the protein fit into this GRAS?
Yeah, they definitely fit into it. I think you just switched to the next slide,
yes.
Right? Yes, indeed. So I don't know if you noticed, but the word safety in the center changed to GRAS. And really the only difference between providing full documentation of safety and GRAS is, again, the public nature, right? The public availability of your safety assessment. Mm-hmm. If you want to tweak the expressed proteins, like the milk and egg proteins, mm-hmm. for flavor or texture purposes, through protein engineering, for example, right? Amino acid changes in the protein. That requires a little bit of extra attention, particularly to address that you don't inadvertently create a food allergen or toxin. Now, it depends really whether this is an issue, how extensive you protein engineer the molecule, but you can easily address this by sequence-based screening methodology, blasting the new sequence against toxin databases and allergen databases. If necessary, if for some reason you have created a new molecule that is somewhat similar to an allergen or toxin, you can then do follow-up work, for example, acid pepsin digestion tests, and if you are able to quickly digest within seconds, actually, in that test, the protein, then the exposure in the gut is going to be really short, and that helps address the issue as well. If that protein is resistant in that test, you can do follow-up studies again, more of a human clinical nature to address the issue.
Would you say that the current regulatory system promotes imitation over innovation, because GRAS seems to be a little bit of a easier pathway to follow than the food additive petition, and there's less burden of research that the company would have to pay to do?
Well, I actually don't think that the use of the GRAS system that we have in the US would be prohibitive to innovation. I don't think it promotes imitation. It's just a little bit easier, right, to convince the regulator that a new substance is safe if it is more similar to something that's already safely used in food. I think we are actually blessed in the US with a food approval system that is science-based, risk-focused, and not relying on concepts used in the EU, such as the precautionary principle, mm-hmm. Which really means you have to eliminate all risk, right, and go for zero risk before the regulators will actually approve a novel substance.
So would you say the US is probably the best place to regulate something?
I'd say the US is a good place to innovate and get cellular egg products approved. And by the way, it's also one of the largest markets. So
Yes.
yeah.
Are there situations where you foresee cellular agriculture products falling into that food additive petition instead of GRAS?
It is possible. So if you create an entirely novel protein that ends up through the initial analysis being somewhat similar to an allergen, then if that requires clinical studies, at that point FDA might advise you to actually file a food additive petition. They will give you the opportunity to retract the GRAS notice. Also, if FDA might have any other questions related to the substance, or if FDA thinks that it can approve your substance but with certain use restrictions. In those cases, they may suggest actually to sort of go back, do some more homework, and either refile a GRAS notice or file a food additive petition. Interestingly, the Impossible Burger, Impossible Foods was mentioned earlier today, and they were in a situation like that where the original GRAS notice was not accepted. They were sent back to the drawing board and had to follow up on addressing allergenicity of the heme protein, the soy heme protein. They are in a position that they can actually refile a new GRAS notice because I believe from what I've seen, they have addressed that issue.
Great.
So even if you end up encountering a roadblock, it doesn't mean the end of the world, and it doesn't necessarily mean you have to file a food additive petition either.
I think the Impossible Burger is a great segue into talking about how we should regulate cultured meat. Maybe first let's talk about how meat is regulated today.
Yeah, so this is where it gets interesting. So with fermentation products, it's easy, it's FDA.
Yes.
Then with cultured meats, one has to distinguish between fish and other meats because it's the USDA that currently regulates meat products, whereas FDA largely regulates fish.
Okay.
So
USDA regulates beef, pork, chicken?
Correct.
Land animals?
Correct.
Okay.
Yep. However, their regulatory oversight currently is quite limited in terms of they look at food safety and hygiene during the processing, the slaughter, right, in the abattoir, and further downstream processing and packing and consumer packaging. That's what they do. They come inspect those facilities. They don't have an approval process, or at least not a formal approval process in place for ingredients. Currently, they actually leverage the FDA in many cases to help them with that process.
Okay.
And so it is conceivable that, or one potential route may indeed be that USDA ends up maintaining the overall oversight over meat products generated through cellular agriculture with input from FDA. However, it really depends on what your product is. If you intend to make and market a steak or a fish fillet, a steak, let's focus on that. Theoretically, the oversight, the final oversight, will be with USDA. However, if through the culture process you produce a protein or a set of proteins that is going to be used as an ingredient, right, in a finished food, then it's actually likely that you can persuade FDA to take full control.
So it seems to me the biggest difference between cellular agriculture products that we talk about and the existing new products that are put through the food additive process are a matter of dosage. Because food additives tend to be additives, just added to the thing, but we're talking about, creating a bunch of proteins that you consume, and that's most of the proteins. Or creating cultured meat, and you consume cultured meat. You don't create a little thing sprinkled into cultured meat. But that leads me to infer that maybe it's better if the first products, the cellular agriculture component is indeed an additive, is indeed actually not most of the product, but just a little sprinkle.
Well, yeah, so interesting you're asking, indeed. it is indeed easier for micro-ingredients to get approval, regardless of which route you follow, simply because to support the safety of a micro-ingredient, essentially all you do is you run a tox study and you show that the safety data allows for a 100-fold margin of safety. Easily obtainable for microingredients. Not so easy to do for a major protein, right? That is going to be a large component of the diet, let alone for a fish fillet or a steak. But on the other hand, it's really the regulatory definition that matters. And food additives are regulated by FDA, whereas meat is regulated essentially by USDA. I don't think you have to go down to small amounts like sprinkles. Even major food ingredients that could make up 20-30% of a food product or even more would still be covered, right, by the food additive regulation and would be eligible for GRAS as well.
What is the maximum percentage of product that you think should be new?
Well, really, as soon as a food product contains more than one ingredient, then each of these ingredients are eligible for GRAS.
Okay. I had another question, which is what existing approved product on the market do you think is most similar to cultured meat?
Interesting. Actually, some of the very traditional fermentation products, which are very solid kind of foods like cheese, they're actually quite similar to cultured meat. There's a combination of science and art involved, right? Just like in cultured meat. They are subject to approval. A lot of the ingredients used and processing aids used to make cheese, they are subject to approval as well. So these would be, and yogurt as well. So lactobacillus, yeast, bacteria, and so on, as well as some of the other ingredients. So it's quite similar.
Okay. What do you think cellular agriculture innovators should think about today to make the regulatory pathway a little bit easier?
Yeah, looking at the state of the art, from what we've heard yesterday, I think there are, it probably is time to start thinking about addressing the safety of your products early on in the process. I'd say you need to be proactive. You need to be creative, but at the same time, you need to be very patient. Expect for this route to be a bit lengthy than you're currently thinking it will be, particularly for the first few products, right, needing to go through the process. But you can help shape it. For your first generation products, make sure that you actually do collect the safety data, tox studies. It is likely going to be required, or at the very least, it's going to be a lot easier to convince the regulators of the safety. If you collect toxicological data, either on your finished product or potentially on some of the key inputs that you might be using. For example, some of the key media nutrients that you might be using that might be of concern. It's a lot easier to actually figure out what the maximum concentration of these ingredients in your finished product might be. You can simulate this, for example, with extensive washing, and then determine what the maximum concentration is, and then design tox studies to satisfy the regulator's concern that might be focused on some very specific ingredients.
Do you think the toxicological data should be collected by the companies themselves or by a contract research organization or someone else?
A very good question. I think the initial research can be done by the company itself, where you figure out what you feel the maximum concentration is going to be based on your process, right? Your optimized process. But when designing the process, keep safety in mind. And if that means including several more washing steps, right, to get rid of some of the ingredients that you've used in the media, for example, do so, and of course figure out how many washing steps it might take, right, to bring down the concentration of anything that might be of concern as well. However, the tox studies themselves, it's advisable to have those run by a contract research organization. They need to be run under good laboratory practices as well for them to be acceptable by the regulators.
I see. Yep. Okay. We have only a few minutes left, so maybe we'll turn on the Slido and get some audience questions in there. I had a question in the meanwhile, which was, do you consider the media that you're feeding the cells to be feed, and is feed regulated?
Yes, animal feed is regulated. I would actually advise everybody to stay away from that concept, because if that concept were accepted, you would actually get involved with FDA Center for Veterinary Medicine. And really, we're not feeding it through the mouth of an animal, so that's the whole point, right, of cellular agriculture. It's actually an industrial process.
Right.
So, but they are considered to be inputs. So they themselves need to be safe and may require approval, specific approval or GRAS status, right, for use in making your product.
I had another question. We met because we were at a panel that was put together by the National Academies of Sciences because they want to change some biotech processes. And to me, the food additive petition of 1958 seems kind of outdated, especially when we consider that there's so many new foods out there, so many biotech products that are not additives, that are not chemical additives anymore. Do you think that the process will change, and how do you think that change affects cellular agriculture?
Yeah, the coordinated oversight over biotechnology may evolve over time. We hope to see more collaboration amongst the agencies, and of course, in this case, we would hope USDA and FDA to collaborate closely or to assign full responsibility to one, such as FDA. Mm-hmm. The food additive amendment is indeed very old. However, and so is the GRAS exemption. But the GRAS exemption actually works very well for industry.
Yes.
Unfortunately, it has come under scrutiny by a number of NGOs who claim that this actually is resulting in a number of quote-unquote secret ingredients in our food chain. Mm-hmm. But I actually think it's scientifically sound, and FDA is managing this program really well. So in general, industry continues to be in support, right, of this program.
Are there any NGOs that are in support of GRAS verbally?
There are, yeah, definitely. Okay. It's not all bad news out there.
Right.
Yep.
I'm going to go to our top question. Do you think Hampton Creek, which wants to get a clean meat product to market next year, is going to face many regulatory hurdles? If so, why or why not?
Yeah, there will be regulatory hurdles. I'm not sure where you are in terms of the regulatory process. Mm-hmm. I'd have to speculate, but if you were to start today and you didn't have solid safety data available yet, then getting to market next year would be a big challenge.
Yes, even in the GRAS process.
Even in the GRAS process, because to run a set of tox studies and get those properly reported under GLP conditions will take about 9 months. Then, of course, you have to compile your dossier, your GRAS determination, have it evaluated by an expert panel. Mm-hmm. All of that takes usually another 6 months. And then, of course, in the case of this type of product, it is highly advisable to file a GRAS notice to establish a precedent. A GRAS notice filing and a review by FDA will take a minimum of 6 months. Again, in this case, probably somewhat longer, up to 12 months. So add it all up, and depending on where you are in the process, if you were to start today with the regulatory process, it will take 2 years.
Okay. You mentioned precedent. Do we need to be working together to make sure that first regulatory approval does well?
I'd say so, yes. And that would be very beneficial. I think this industry needs to organize itself. It has benefited the enzyme industry tremendously. It is benefiting the broader biotech industry tremendously. Not sure if you need to organize as a cellular ag industry or become part of a larger movement. You could think of your startup companies joining the Bio Trade Association, which covers all of biotechnology. That's an option as well. You need a strong voice in Washington, particularly again when it comes to cultured meat products that potentially will be regulated by 2 agencies.
Yes, of course. Another top question, are there countries where the regulation would already allow the sale of cultured meat today? There
might be a few. However, it is my opinion and that of most self-respecting and responsible companies to fully evaluate the safety first, establish GRAS status, and once you have GRAS status, then take it to other markets that do not have a formal regulatory approval.
That leads me to another question, which is, do the different regulatory agencies in different countries swap notes?
They do at a high level, but when it comes to individual approvals, for example, based on the submissions you make, they generally don't.
Okay.
And they can have very different approaches, like in the EU, where they use the precautionary principle. They don't even want to see a GRAS notice, even though it's already out there published on the website. They don't want to be, quote unquote, tainted by that.
Do you feel comfortable talking about labeling?
Sure, absolutely.
What labels will cellular agriculture require that other foods might not, and vice versa?
Well, it for one, is going to depend on how you want to market the product, or how potentially a customer of yours might want to market the product. Is it going to be meat? In that case, USDA will get involved, and it will want to see full substantiation, right, of equivalence of your product with actual meat from an animal. Is it going to be? Is it going to have a qualifier? Is that going to be used, like cultured meat, right, or meat analog, or something like that? So it may indeed be that the product label will have a distinguishing feature. Will there ever be anything else that's needed on the label, like GMO, right, or non-GMO? Yeah, it depends, of course, how the product is made. If you're using yeast or another microbe, right, that is genetically engineered, and the resulting protein is part of the food, then you will be subject to GMO labeling at some point.
We only have time for one more question. I wanted to ask you which cellular agriculture product you think should be the first one to be in the regulatory pathway.
Should be or will be?
Should be.
Should be.
Strategically thinking.
Yeah, so I think the fermentation-based products are just going to have a relatively easy route.
The milk and egg proteins.
Yes, getting approval for them will be exciting, but it actually will not really provide a good precedent for cultured meat.
Okay.
So, because that will be looked at as a separate case, a standalone case.
Okay.
So getting one of the cultured meats through the regulatory process will be fantastic.
And so then I'll change my question to, what should the first cultured meat product look like that gets regulated? Strategically speaking, for the easiest approval that sets the best precedent.
I see. Well, so regardless of where individual companies might be in their process and so on and so forth, so I would actually think that a cultured fish product that is going to be marketed as an ingredient, right, would be a good one. Why? Because one, it's an ingredient, so as such it would qualify for the GRAS route, right? And two, it's fish, which is exclusively regulated by FDA, so you won't have the gap to deal with. And you set the precedent
I thought fish was USDA.
Nope, fish is FDA.
Oh.
Yeah, meats are USDA,
Okay.
Or at least meat processing is. So, of course, all of that is irrespective of where folks are in their development process. But ideally, if I had to choose, I would go with a fish ingredient, get that through FDA, get them comfortable, right, with the process methodology and so on and so forth. One thing to emphasize here, though, is I think regardless of which product one chooses to or would choose, right, to take through FDA, establish a really solid safety evaluation methodology, a framework for safety evaluation that is going to be applicable not only for, in this case, fish, if that were the first one, but for all cultured meats, and get some expertise behind it, right? Some recognized names of food safety experts.
Okay, great. Thank you so much, Vince. We'd love to give him a round of applause.